basic science and translation. You know, the previous speaker described a phenomenal intervention, but every day I went to my hotel room and thought, oh my God, I saw this one molecule or this one target and it will change the world. I mean, so I really get very enthusiastic about everything that's in the pipeline, you know. And I'll talk about methylation studies, but because I'm the last speaker, I feel I need to summarize some of my impressions, which are totally biased. But we saw many talks on biomarkers of aging. For example, Vadim Gladyshev just presented this phenomenal talk on transcriptomic biomarkers and other biomarkers. But there were also talks from Max Unfried, Brian Kennedy, describing lipidomic and metabolomic clocks. Of course, we know there are proteomics clocks and endless methylation clocks. As many people have pointed out, or I should say, probably your head is spinning. There's so many clocks. What should we use, you know? And the great advantage of a proteomic clock or metabolomic clock and so on is interpretability. If you really care about that, inflammation, I want to measure inflammation, use an inflammation. So the other thing is, if you really want to predict future morbidity, then use clinical biomarkers that detect early morbidity, right? So I have this tautology, biomarkers that measure early morbidity, let's say blood pressure, are more predictive of future morbidity than an omics clock that measures accumulated damage, right? So having said this, the aging field is very interested in biomarkers that measure accumulated damage. And I think it has a lot to do with our new understanding of the nature of aging. It's really accumulation of damage and dysregulation. I want to start out with the main weaknesses of methylation clocks. It is this issue of biologic interpretability. I mean, one can make methylation clocks interpretable by focusing on, for example, PRC2 methylation. Vittorio talked about that. So you can focus on methylation in certain chromatin regions. So that's a weakness. But we heard phenomenal talks by Raghav Sehgal and Ryan Smith that showed that these methylation clocks and methylation biomarkers are already being used in human clinical trials. And I think the talk from Ryan Smith explains why methylation is such a strong predictor of future morbidity. And that's, for me, the key question. Why is it that these methylation clocks work so well? Why is it that you take somebody's blood samples and you can predict time to death and so on, you know? What's the mechanism? Then I think the field, of course, the biomarker field, needs to integrate all these different layers of the biological hierarchy. You know, I think that will come. We need longitudinal studies in humans where we have methylation data, proteome data, metabolome data, and all sorts of omics. Once you have longitudinal data with outcomes, you can build wonderful models. And we heard talks from Rafa De Cabo and others that describe these enterprises of collecting data. But I want to come back to strengths of methylation, despite all the limitations. I think people like methylation clocks because of an intuitive appeal, because it relates to a molecule that carries genetic information. We now know that this DNA molecule is also a clock. That's just intuitively very appealing. And also there's this intuition of the fact that the aging process is innate, it's universal. everything ages, all mammalian species age, all tissues age. And so having universal multi-species methylation clocks or pan-tissue clocks appeal to that. And then the fact, so another reason why they are so intuitive is that methylation clocks are life course clocks. You really can measure time from prenatal samples to centenarians. And Vadim Gladyshev just showed you how methylation clocks indicate a very early rejuvenation process. That's a miracle of methylation clocks, that developmental processes are linked to aging much later in life. So this is the intuitive appeal, but there are technical advantages. First of all, I would argue methylation clocks are pretty much ready for human clinical trials. Many of you are already using these clocks. Clearly, there will be better and better versions. There were many talks by different people who describe new versions of epigenetic clocks. And we will at some point have to have an unbiased look and evaluation of these clocks, maybe as part of these biomarker consortia that Vadim Gladyshev and Jesse and Mahdi Mokri organize. There's another technical advantage of methylation clocks. They actually work in vitro studies, you know. I mean, clearly, I love it that people use them in human clinical trials, but the very same clocks can be used for studies, for studying cells in a dish. So Vittorio Sebastiano showed how OSKM rejuvenates fibroblasts and other cells. And then my favorite reason for methylation is the ease of developing pan-mammalian clocks. And there was this wonderful talk from Jesse Pogani and the Gladyshev lab on heterochronic parabiosis. We really learned so much from this paradigm, but what maybe escaped many audience members is that Jesse used pan-mammalian clocks. In other words, whatever moved this clock in the mouse model probably will also work in humans. Why? It's a pan-mammalian clock. So I'm talking about pan-mammalian clocks. I call these multi-species clocks third-generation clocks. And one way to build a third-generation clock is to predict what I call relative age. So what is relative? Relative to what? Relative to maximum lifespan. So if you are a 61-year-old human, then your relative age is 61 divided by 122, which is the maximum lifespan of humans. So a 61-year-old human has a relative age of 0.5. And similarly, you can measure relative age in all species. And the miracle is how easy one can estimate relative age with methylation. And this brings me to another set of phenomenal talks. Jackie and other speakers described facial clocks, extremely appealing. And Alex was one of the first people to develop such a clock. And I remember when I met Alex, he had an app on his phone, and it nailed my age up to 1 or 2 years, you know. So it's such an appealing thing. But my challenge to the AI team is develop a facial clock for multiple species, you know. So, start out with linking a human with a whale face, then go to the dog and then a mouse. And after you succeeded at that, then put in the face of the axolotl, you know. And, and speaking of which, there was a talk by Julia Halusa, Joseph Zoller, Max Yun, who did describe these dual species clocks that measure age, relative age really, for axolotls and frogs, so amphibians, but also stunningly humans and amphibians, you know. So, and that to me is yet another miracle of cytosine methylation. It really speaks to my claim that there's this innate aging process that can be observed really in so many different species, certainly vertebrates. So going back in time, when I developed the pan-tissue clock, I wanted to find an intervention that actually resets it. And the only thing I could find in 2013 was the Yamanaka factors. iPS cells were perfectly young, according to the pan-tissue clock. And this finding is a gold standard. iPS cells always are young, according to all epigenetic clocks, including pan-mammalian clock. So that's established. But the field needs to sort out the issue about partial or interrupted reprogramming, because there things are much less clear. For example, there were great talks by Vittorio Sebastiano, David Sinclair, Luca Kulik, Boris Georgievich. Many companies right now pursue that idea, but we need to figure out what dosage, what kind of factors, and in what tissues do we observe epigenetic rejuvenation. So partial programming has a weaker signal. So now I want to move on to a different concept, maximum lifespan of species. Let's not talk about aging per se. So most people, including myself, really confuse species characteristics with individual characteristics. So maximum lifespan is a species characteristic. Maximum lifespan of humans, 122 years. Maximum lifespan of a rat, 3.8 years. That's a species characteristic. However, we, as individuals, we actually only care about one characteristic, time to death or time to morbidity. That's or your age or your biologic age. These are individual characteristics. And I do want to tell you, mathematically, these two concepts are completely different, but it turns out also biologically they're very different. And the best example to illustrate it is the oldest human who ever lived, Jeanne Calment, famously was a smoker. So if you just copy her, whatever she did, you know, what I want to say is smoking doesn't affect the maximum lifespan of our species, okay? Really doesn't. Of course, it will affect your time to death, you know. And however, the longevity field has long been fascinated by molecular determinants of maximum lifespan in species. And we heard phenomenal talks by Domenico Di Fraia, Vera Gorbunova, Jan Wiek. Many people among the audience have studied maximum lifespan. And me too, you know, I was fascinated by it. And so I assembled a team, the Mammalian Methylation Consortium, 200 investigators. Many people in the audience can find their name here. Vera Gorbunova, Vadim Gladyshev, Joe Takahashi, many of you are among the author list. But together we assembled this large data set, which is publicly available, 348 species of different lifespans. And so what were the key insights after 7 years of pain? And here they are. Biologic processes that relate to time to death of an individual species. What is that? Caloric restriction in a mouse, smoking in a human, obesity. All of these things that affect your average health or lifespan, they typically are completely different from the pathways that determine maximum lifespan of the species. So biologic age that everyone talks about is truly an individual property. And so I had this question, did I just waste 7 years of my precious life to assemble a data set and it doesn't inform drug development at all? And there was the best talk of the entire conference, and I want to say the best talk ever in the longevity field, by Alex Zavoronkov, who coupled drama of science, I could barely contain my excitement, with this amazing romantic twist at the end. So I invite you, the eternal longevity research partners, Dominika Wilczok and Alex. Anyways, I want to, many people ask me about the mammalian methylation platform and mammalian methylation studies. And I want to tell you, I started a non-for-profit company which offers mammalian methylation clock testing. It's a fee-for-service arrangement. Anybody can interact with them. It has nothing to do with my current employer, Altos Lab. So I personally like the mammalian methylation array for technical reasons, but also all of our software is freely available for that. Anyway, I want to talk a bit about some of the surprises of studying maximum lifespan with methylation. One surprise to me was that when you do an upstream regulator analysis of these lifespan-related cytosines, you do see OCT4 and SOX2 and some pluripotency genes. Why is that interesting? Because it's, again, these Yamanaka factors. That biology, strangely, the biology of reprogramming seems to be linked to the biology of maximum lifespan. Interesting to me. More generally, you could ask, what is the methylome of a long-lived species? And here I show you in red the curve, the methylation landscape on the DNA of a long-lived species. It's hilly. Low methylation at transcriptional start sites, high methylation in gene bodies, high methylation in intergenic regions. So hilly landscape. Long-lived species. By contrast, a short-lived animal has a flatter landscape. And I think it has a lot to do with gestation time. These animals are poorly built. The methylation landscape is poorly built and poorly maintained. So coming to another paper, predictor of maximum lifespan. We literally have a multivariate regression model that uses a couple of hundred cytosines, and that predicts maximum lifespan on a log scale. And so then this is a biomarker, and personally I think it's not a useful biomarker, but others may disagree. But you can ask the questions, which interventions affect the predicted value of the species' maximum lifespan? And I have the caution here, this is about a species characteristic. Probably it doesn't matter for you and me. In any event, the first thing I looked at is, of course, interrupted epigenetic reprogramming, right? And there's no effect, So this interrupted cyclic reprogramming did not affect the epigenetic estimate of the maximum lifespan of the species. It may still affect the average life expectancy, and I was so happy that several speakers talked about mouse studies where they do lifespan studies of transient reprogramming. I briefly want to mention we also have predicted maximum lifespan for human cohort studies, and our species characteristic does not predict individual mortality risk in any of the cohorts we looked at. Another thing you can ask is, what about cytosines that correlate with maximum lifespan? Do they correlate with chronologic age? And the answer, it should be, right? Something that relates to maximum lifespan should change with chronologic age. But exactly the opposite is the case. So there is barely any overlap between that. So I cannot highlight that enough. There's a distinct CPG pattern. CPGs linked to chronologic age do not relate to CpGs linked to time to death in humans or in mice. And this is deeply frustrating to me, this finding. And so the hope is, of course, well, what about if we form some sort of Venn diagram? Or what can we do? And that brings me to a paper that will come out in a week or so, which is about methylation dynamics, velocity, gradient, or whatever you call it. I call it the rate of change in methylation. So imagine you have a longitudinal study. You measure blood methylation from age 0 to age 100, and you calculate a rate of change in methylation. That rate of change will turn out to have a very significant correlation with lifespan. If you look at the right CpGs. And so this plot is a little bit hard to read, but each dot corresponds to a different chromatin regions, promoters, heterochromatin, enhancers, gene bodies, flank, blah, blah, blah. And the right-hand side shows you chromatin regions where the rate of change of methylation relates to 1 over lifespan. And it is these bivalent promoter regions and repressed by PRC2 complex. But the reason why I show this graph is there are many regions on the DNA that don't show that pattern. So you need to think about it, how to define it. Anyway, here's the graph. On the y-axis, you see log-transformed rate of change of methylation. Every dot is a species. And on the x-axis, you see log-transformed maximum lifespan. Beautiful relationship. And you've seen this type of relationship a lot in your life. How often have you seen it? 10 times. That's an old relationship. People have shown the same for somatic mutations, for telomere length, for repair. Probably you can think of other things. That should make you very nervous that you've seen it. And one of my punchlines is, if you have a biomarker that correlates with age, it will show this relationship. I'll come to that later. Anyways, let's talk about the dynamics of methylation changes across the lifespan. So by having the data from the mammalian consortium, we can look from cradle to grave in pigs here. And there's a distinctly non-linear relationship of gain of methylation at these regions. And more, maybe you appreciate it from here, the rate of change in young animals is much steeper than in old animals. And that bears out. So, but interestingly, the rate of change in young correlates with the rate of change in old animals. I find that a high correlation. Speaking about correlations, there's another interesting finding for the quantitative types. Here I show how chronologic age correlates with methylation in different species. Each dot is a different species. And on the x-axis we have lifespan, and there's no relationship. So if I was a physicist, I would say, well, maybe this correlation of age with methylation is a species invariant, which explains another mystery of methylation. Why can you build epigenetic clocks that are equally accurate in a short-lived species like a mouse as in a long-lived species like humans? Why is it always the same accuracy? Well, this has a lot to do with that. So these fundamental equations that result from our methylation studies in mammals lead to several main insights. The rate of change is inversely related to maximum lifespan in bivalent regions, but not in other chromatin states. The correlation between methylation and age bears no relationship to maximum lifespan, and the rate of change in young animals is correlated to the rate of change in old animals. So now I'm speaking to editors of journals and reviewers. Because coming back to my statement, any biomarker that increases with chronologic age will result in a strong inverse correlation between the rate of change and maximum lifespan. And this is just the trivial consequence of the definition of the rate of change. So one can derive, the bottom shows you a mathematical correlation property where I didn't use any data. This is math. Rate of change is 1 over lifespan times some factors. So one really needs to address this bias in the definition. And I want to end by thanking my many collaborators, Vadim Gladyshev, Vera, Max Yuan, Christoph Niers, Jesse Pogani, Joe Takahashi, and all the people from the Mammalian Consortium. Thank you so much.
Thank you. Thank you. Thank you so much, Steve. That was a fantastic talk. Really amazing. We have time for a few questions. I will start with asking a question because I have the microphone in my hand.
Okay.
So have you looked deeper into the chromatin? Like you could predict secondary DNA structures or something like that, and whether or not there are changes in DNA methylation in those areas?
Yeah, I think many people look at that, and some talks talked about these R loops. I think that I personally don't have anything to report on it, but my instincts say that there is a connection. Yeah.
Very exciting.
Mm-hmm.
Steve, amazing talk. Rate of change, I like the last slide that you had. I kind of lost myself on
Which
it.
one?
It's a little profound. Could you explain it again?
Which one?
This slide. Can you explain specifically your last equation a little more? It's a little complex, and I want to understand it. And I think maybe a few others in the audience
This is
will.
now math, and then
I know.
people will guzzle down extra liquor to get it out of their system, but okay. So assume you want to calculate a velocity, a rate of change. The upper equation shows you that. You form a univariate regression model where your biomarker gets regressed on age. The rate of change is the beta. The beta is the left-hand side of the equation. So far, so good. Then there is that rate of change can be actually expressed in a beautiful formula, I think, which is that it's just the Pearson correlation between methylation and age divided by the standard deviation of relative age. Do you remember I talked about relative age? It's 1 over lifespan. And that's it. That's the equation. That's a little homework exercise. But I think, I hope that people do this exercise, preferably today, because I'm really tired of reading papers on this biomarker relates to maximum lifespan. I'm just tired of it. I think the reviewers need to ask tougher questions and say, all right, do some proper permutation tests, control for it, Because again, this is without any data, Mm-hmm.
Yeah. So thanks. Does this trend?
Oh.
Yeah. So this connection between maximum lifespan and time to death for epigenetic data, do you also expect it for transcriptomic and proteomic data?
Sorry, whether we can build time to death estimators?
Yeah, you have this connection between maximum lifespan and time to death in terms of the markers of epigenetics.
Vadim should answer that, I don't know. I just think we need to be open to the idea that it could be difficult to translate interventions that enhance maximum lifespan for us, for health span purposes. Does that make sense? It could be that this is really the goal of ARDD2024 in 100 years, where they have species interventions. But yeah.
Yeah. I have a question regarding why do you think it's specifically the bivalent promoters that are correlated with maximum lifespan?
Oh,
But it's a biology question.
Yeah, that's an easy answer. So these bivalent regulatory regions that are typically bound by the PRC2 comps, polycomb repressive complex 2, they exhibit low methylation in pretty much all tissues, but also all mammalian species. And that part of the biology is one of the reasons why you can build pan-tissue clocks, pan-mammalian clocks, you know. So the polycomb repressive complex represses, but it does it without methylation, you know. And so yeah, then you have low methylation, and the only thing you can do with aging is to go up. Do you see? So coming back to whatever goes up with aging will then have this inverse relationship to max lifespan. Mm-hmm.
Yeah. Thank you, Steve. Here, I have a question. Steve, here.
Yeah, okay.
Thank you. It seems that there's something magic to DNA methylation to predict a biological age. Is any connection with the heterochromatin transposon silencing, chromatin organization? Because this is the function that DNA methylation does at the end, right? Maybe there are some changes there. Can you speculate something? Have you seen something?
Oh yeah, I mean there are now several groups have, for example, developed epigenetic clocks that measure methylation in transposable elements, line 1. I mean Vera has such a clock, but a couple of people. And methylation, I mean the answer is there is, of course, the closest connection, you know. So, and people study it. Methylation in enhancer regions has effects, you know. So everything is connected. Coming back to actually the advantage of these black box pan-tissue clocks, because if you allow a mathematical optimization method to capture it, you probably capture a lot of these chromatin changes, you know. If you go in a completely agnostic way, you capture it all, you know. But then you don't have interpretability, you know. So the opposite would be build a clock that only uses line 1 methylation, or only PRC2 methylation, or only enhancer methylation, you know. So yeah. Mm-hmm.
Hi, Steve. Great talk.
Thank you.
I have a bit of a weird question. So I heard Jennifer Doudna discuss when she discovered CRISPR that she was excited, but also kind of scared about the implications of her discovery. And, you know, we can now use these clocks to predict, for example, when people will die, like with GrimAge. Is there any part of this that has scared you or that you're anxious about regarding these discoveries?
No, I'm zero fears about biomarkers. Yeah. Because think about it, blood pressure is being used every day. It's very predictive of mortality risk, you know, and people live with it. Yeah.
Hi, Steve.
Yeah.
Great talk.
Yeah.
So I have a question about, of course, partial reprogramming.
Yes.
So you've shown that the current clocks don't really work so well with partial reprogramming, and thus the slope does not change in the maximum lifespan clock, right? So
Yeah, I wouldn't quite put it this way, but
okay,
maybe finish your thoughts. Yeah.
Okay, feel free to correct me.
Yeah.
Okay, so I'm just wondering if you think this might be due to the fact that maybe the partial reprogramming protocols that were used were not as optimized to detect such changes. So
yeah, for sure.
Okay,
Yeah. So let me rephrase things a bit. So so when you look at partial reprogramming long enough, you know, at some point you see the rejuvenation. How many days do you need? You know, that's the question. But I agree with you. It's not optimized on many levels, you know. So probably that's one of the reasons. It really also depends on tissue type, you know. What I showed you, though, was a different question. So I had an epigenetic estimator of maximum lifespan, not average lifespan, big difference. So the species characteristic, I couldn't see an effect, which is unfortunate. I would have loved to see one, but I didn't, you know. Yeah. Yes.
Hi, Steve. It's a very fascinating talk. I'm Christine. I'm a longevity physician, so I'm always dealing with human beings. And the rate of aging is quite a fascinating idea. And I'm wondering, for as long as we want to calculate the rate, how long should be the time interval for the test?
Yeah, that's a question. So there are so many commercial companies and wellness clinics, and they offer biomarker testing. And the question is, how often should you measure methylation? I would not measure it more often than every 6 months, possibly once a year. That's my opinion. But I hope somebody will prove me wrong, and then soon we have a smart watch, and then every minute of the day you see methylation changes. And I want to finish with one thing. There was a study that claims that there's a circadian rhythm, talking about Joe Takahashi's work. So apparently, if you measure your epigenetic age at midnight, you're slightly younger than at lunchtime. So if you want to game the system, you know when to draw your blood.
That's why it's a great idea to go to bar 7.
Yeah. Okay. Thank you so much.
Thank you so much, Steve. Amazing.
Thank you.